Okay, this showed you how those enzymes, glycosylase enzymes, can recognize the base, mismatched base, because it's not making the correct hydrogen bond. So this base can be hanging outside of the double helical structure. That's how it can easily recognize and cut. Okay, so a kind of three-dimensional model of the enzyme activity. So if damaged base is not removed by base excision before DNA replication, another the glycosylase enzyme called a fail-safe glycosylase can remove that mismatched nucleotide and incorporate a correct one. So there's a double layer of a repair system in place. That's what it's saying. Okay, so base excision repair can do that, fix that. And then maybe oxidation could do the damage. If this will be used without being fixed for the replication, then during this time, I guess the mutation will take place. But before that, there's another glycosylase that can fix that problem. That's what is shown up there. Okay. Here's another kind of excision repair. So surgically repairing the damaged part. That's made up with multiple enzymatic complexes. Okay, so especially when dealing with the UV damage, these enzymes are involved. For example, let's say DNA is exposed to strong UV light. You see something like thymine dimers and things like that happen. So DNA structure will be damaged because of unusual covalent bond between the bases and things like that. So if that happens, okay, these set of enzymes are made available under the stressful condition. Yeah, the genes that are coding for these enzymes are turned on. We'll talk about the SOS response as part of that response. So anyway, so these enzymes are made, and then they come along available, and they make the repair. So the way they carry out their task is, first these two dimers of these complex UVRA, UBLB, they bind around the damaged DNA area because they are distorted, kind of with a very strange structure. So they come along and bind, okay, around the distorted DNA helix. Okay, and then upon detecting a distorted DNA, UVA, okay, once they recognize there is a problem, and once they bind, and then UVA, those two proteins leave the UBB alone. Okay, and then UBB recruits another protein called UBC. Okay, now UBC comes along and creates two incisions, so UVC can cut, okay, on a single strand around the distorted area. Incision is made. It's about 3' end 4 to 5 nucleotide away from the distorted area, and about 5 to 8 nucleotide away from the incision area. Okay, so yeah, 3 or 4 away on the 3' end, and the 5 or 8 away on the 5' end. Okay, so this part is cut off. Okay, and then UVD, another protein is helicase that will come along and removes by relieving and getting rid of the hydrogen bonds. So this part is removed. Okay, and then this gap is now filled by DNA polymerase 1. That's a repair enzyme we talked about in the E. coli system. And then once it's filled again, DNA ligase will come along and fill the last phosphodiester bond. Okay, so this is a very well-known characterized repair enzyme system, so UVR system in E. coli cells. Okay, in higher, I guess, living system like human, there are similar systems in place and similar enzymes. And then if there's a problem in any of those enzymes, in the human case, maybe there's a mutation, so any of these enzymes are not made, then you can come up with some kind of genetic disorder. Okay, we call those genetic disorder as xeroderma pigmentosum. Okay, so that's the genetic disorder where these people with certain mutation, they don't have these enzymes made in their system. So, if they're exposed, then there's a higher chance that the DNA will be UV damaged, and then those damaged DNA molecules are not repaired. So then they will have higher rate of skin cancer, okay, and lesion and cancer. So that's what they have. Okay, so we can understand that. Another excision repair mechanism is associated with RNA polymerase enzyme. So RNA polymerase, it does what? Transcription, right? It is making RNA copy of DNA. So let's say somehow that the damaged DNA is not corrected, repaired before there's a gene expression. Okay, so somehow that gene has to be expressed under this condition, whatever this condition is. Okay, so RNA polymerase was engaged in the promoter sequence. Now it's reading through the gene sequence area. So RNA is being made through this transcriptional activity, right? So along the way, when RNA polymerase moves along the gene sequence trying to transcribe, okay, then when RNA polymerase faces that distorted DNA, okay, it stalls. Okay, it stops the transcription, it kind of waits, and then it doesn't know how to handle that, right? So then it kind of folds up, okay, and then the stalling of RNA polymerase recruits, okay, those repair enzymes, those excision repair enzymes, and instead they come along, and after dissociation of RNA polymerase, they bind around the area, so they'll do the repair. So it's a way of not transcribing and maybe translating distorted, mutated gene, so you'll make a faulty protein from this gene. Okay, so that's a kind of protection against that possibility. Okay. Okay, among different DNA damages, we mentioned about DNA can be damaged through maybe breakage on its strand, maybe nicking, meaning one of the two strands are cut, or cutting of both strands. Okay, so what if you have damage on two strands, maybe both strands are cut, or maybe one of the two strands are cut? How do you deal with those damages, right? For example, if you have double strand break, okay, that there's a repair pathway, repair enzymes that can take care of that repair by retrieving the sequence information from the sister chromosome to make recombination repair. So recombination repair is between two different sister chromosomes. The similar segment can be recombined, okay, to make the repair. That's what it means. Okay, so if there is the replication going on, there's another sister chromatids with the same DNA sequence available, the recombination repair is what's going to happen. That's what it means. Okay, and then DNA polymerase can stop around attempting to replicate over the lesion, damaged area in a replication fork, encounters a lesion in DNA. So we're talking about this. Let's say that this is a DNA template that is just being replicated. So the replication fork is going on, right? So what if there is a nick on the template strand, one of the two strands? Okay, so the bottom strand there's no problem, right? But the top strand, okay, when it goes continuously, so the nick, okay, at that spot, okay, so the template DNA is damaged. So polymerase cannot go any further, so it's going to stop right there. So then double strand, double strand break for recombination. So since this is much shorter, okay, now there should be a recombination repair that should take place there. So maybe you can use these as the copy, and then you can make extra copy and then put those along there. Okay, one of the possibilities is that there's a lesion instead of a cut. There's damage there. Okay, again, bottom strand is fine, but top strand there's a lesion, so it cannot go any further than lesion area. So what's going to happen is there's a unique thing happening here. So between the newly made strand, so in this case, this strand should have the same sequence as that strand, because so this newly made strand is a copy of this bottom strand, which is complementary to that. Okay, so actually this strand should be same as that strand. Okay, so at this place, there's going to be unique kind of conformation change made. So this DNA newly made strand is now folded certain way, so it can be used as a template to make the DNA copy over the lesion area. Okay, so that's how the DNA polymerase can bypass and continue the polymerization. Okay, so it's kind of unique repair, kind of complicated repair enzyme, and how can bypass that area during the DNA replication process. Okay. Okay, so So double-strand breaks are also repaired by direct joining broken ends. So what if you have, instead of nick, what if you have both strands are cut? It's a double-strand break. Then what happens? So that break area can be repaired by a bunch of these enzymes. That's what it means. So double-strand breaks are mostly cytotoxic. It's very dangerous for the cell, okay, because these damage can be degraded. So the DNA damaged and degraded, and then there's no DNA, then cells will die. So that's what cytotoxic means, because it causes replication block. So if DNA is not synthesized, then there's no replication, no cell division, right? And then chromosome may get lost, and which will cause a cell death. So it's very dangerous for the cell. Okay. So then, if you have those kind of double-strand break damage, then cells have multiple pathways to deal with those damage. Okay, so, and among those different mechanisms in yeast, the fungi, unicellular fungi, there are some process called recombination base double-strand break repair. That's a long name, but RBDS, okay, PP, recombination based double-strand break repair system can handle most of double-strand breaks. But if the damages were made before the sister chromosomes are available, because sister chromosome can be used for the recombination repair, right? But then what is it going to do? It's going to just repair right there. So there is going to be non-homologous end joining system, okay, and non-homologous end joining systems. So what is described is non-homologous end joining system, enzymatic system, that can join through base pairing between the two ends. Okay, so what it means is, the broken end can be joined up together. Okay, it's like a soldering. Okay, and that's yeast. And then in mammalian cells, okay, what's shown is the mammalian system actually Ku70, 80 proteins and DNA PKCs and Artemis and XLCC4. I mean, those guys, okay, can come along, okay, and then do the sealing of those two broken double strand, okay, and take care of the problem. Okay, that's what is shown up there. So double strand breaks are also sealed. So that's the main message there. Okay, and then highly error-prone translesion synthesis can be sometimes kind of beneficial for the cells, especially if the cell is under a lot of stress. Okay, so what does that mean? Highly error-prone translesion synthesis. So what that means is DNA polymerase is making a bunch of mistakes. That's what that is, highly error-prone translesion synthesis. So DNA polymerase replication is going on, but that the replication is very faulty. So it's going to make a lot of mistakes. A lot of mistakes means what? It's a lot of mutations, right? So creating a lot of mutation may be beneficial for the cells. That's what it means. Okay. By allowing a DNA replication bypassing DNA damage. Okay, so what it means is, let's say there's DNA damage. So if these damages are there, and what are you going to do? I mean, during the replication process, right? If it's repaired back and become normal, that's fine. But if that they're not, repaired, then what happens? Okay, so what's going to happen is there are specialized DNA polymerases, okay, such as the Y family DNA polymerase, or DNA polymerase number 4 or number 5. Okay, those special DNA polymerases can read over the area where the DNA damage is on the template DNA. Okay, so by doing so, they make low fidelity DNA synthesis. What that means is, the replication doesn't have to be perfect. Okay, G to C, A to T matches. What they're making is not perfect, okay, but they can move along that damaged DNA area, so the replication can continue. That's what it means. Okay, so what's the point of that? What the point of that is, under the stressful condition, normally cells going through a lot of stress, maybe it's very mutagenic, but the replication should take place, so the cell can divide to make daughter cells, right? So then, how is it possible? Normal DNA polymerase cannot move along over this damaged area, but instead, under the condition, instead of using normal DNA polymerase number 3, these polymerases will take over. Polymerase number 4 and 5, they can move along that area, but instead, although they can read all over this area and continue their replication is not going to be perfect. That's what it means. So by replicating, yeah,